Hey guys, this is Mrs. Holland Chambers with Holland Chambers Biology, bringing to you the pickle experiment. Now the pickle experiment is an introductory lab that's going to walk us through the scientific steps of how to do an experiment. And so what I want you guys to do right now is take out a sheet of paper. You're going to be writing down all this information that we're going to be going through, including the data, and your job at the end of this, which I will assign in about a week, is to then write a formal lab write-up. So you're going to type up all this information that we're going to be learning in the end. Okay, so that's going to be our final project with this. Now for right now, just pencil and paper, write this stuff down. That's all you need. Okay, so pickles, right? Who doesn't love pickles? Well, I observed while I was shopping that there's a couple different types of pickles, right? There's butter chip, there's regular like dill pickles, there's salted, there's all sorts of varieties. Well, I decided to try the butter chip pickles, all right? And I thought, wow, these were amazing. They're sweet, they're already cut in like little tiny, circles, they're easy to eat. And I was thinking to myself, wow, if these are so amazing, can I make them even better than this? Well, I just walked you guys through the first two steps of the scientific method. Observation, problem. So the first thing we do as scientists is we observe what's going on. Well, there's a bunch of varieties of pickles. And there happens to be this new pickle that I didn't know about, which is the butter chip pickle. Now, my problem, my problem is, would adding salt or sugar make them better? So I want you guys to stop the video right now and write down the observation and problem. So OP, observation is that I noticed the butter chip pickle was better than regular pickles. Problem is, would adding salt or sugar make them better? Well, before I just jump into an experiment, which you guys are going to see me do here in a second, you need to do some background research as far as, has this been done before, what's going on? Because if you guys can just Google the problem and there's an answer, why are you wasting your time doing this experiment? You guys hear me? Okay, so background information is I. So I information. So I'm going to do some background information. I'm going to see if there's any of these different types of pickles out there. So are they chocolate pickles, extra sugary pickles, extra salty pickles? And what I'm finding is that the ingredients in the pickles themselves is a lot of them contain sugar and salt. So that's a pretty, that's a thing that's going on. So now that I did some little education background education on my pickles, I'm now going to formulate a hypothesis. Now let me take a step back here for a second. Most of you guys have been trained ever since elementary school that a hypothesis about these pickles is an educated guess. And I'm going to have to tell you guys, and I'm really sorry, but that's wrong, okay? So wipe it out of your head. It's not an educated guess. That's completely wrong. What a hypothesis is a testable prediction. So I need to make a claim, a hypothesis that I can actually test so that way I can come up with a conclusion on my problem, okay? So it's not just a random guess, it's a testable prediction. So do me a favor, you're going to stop the video here in a second, and I want you guys to make a claim. What is your testable hypothesis? And I want you to start it off with, it is believed. So it is believed that the sugary pickles will be the best. It is believed that the chocolate pickles will be the best. It is believed that what will be better because. Okay, so why are you saying chocolate's going to be the best pickles, and why are you saying sugar or salt's going to be the best? Now keep in mind our three choices that we're going to have is chocolate syrup, super sugary, and why not? Everybody loves chocolate. You have sugar, okay, and then we also have just good old salt, all right? So make your testable prediction now. the experiment. And so, here you go. Pickle experiment commencing now. All right, guys, so control, grab your pickle and go. And again, it's whatever your personal taste is. You don't have to tell anybody else, so make your mark. All right, next up, person B. All right, person C. Make your mark. Okay, person D. Is this your personal opinion? Yes, Sarah, you're right. Over here. All right, person E. Okay, so again, our problem is with adding sugar and salt to butter chip pickles to make them taste even better. So next up, guys, is your sugar. So now we're going to try butter chip pickles with sugar. So if we add sugar, do they taste better or worse? So again, butter chip pickles are awesome the way they are, but can we make them better? All right, next up, person D. Okay, make your mark. Again, 1 is horrible, 2 is okay, 3 is interesting, and 4 is awesome. Okay, so butter chip pickles are a fermented cucumber, so if we add more salt to it, let's see if it tastes better. Okay, so next up is our salt. So adding salt to pickles, butter chip pickles, doesn't make it good or bad.
Ready to run? Yep.
Alright, you like them all? Alright guys, last up. We added sugar, we added salt, so now let's like make it super sugary and add chocolate because who doesn't like chocolate? So what is your rate of taste? Butter chip pickles and chocolate. Feel free to face the camera with those reactions. Alright, so person C. Alright guys. What do you think? You can't go wrong with chocolate. This thing's over there, guys.
I'm terrible.
Okay, now everyone's done with the experiment. We had 5 different people run this experiment for us. And so they were able to check, based on their own personal preference, what their rate of taste was, okay? Now please keep in mind that this was different ages. They were different gender. You know, maybe they had different backgrounds. Maybe one was raised Norwegian on fish and the other one was raised straight up American on A's burgers, right? So, you know, what's your taste preference? What's your background? Will that influence or, you know, affect the data that we got, all right? So kind of make sure you guys think about that. Also think about the data that was collected here. Were all the pickles the same size? No, I just cut them up real quick. Did every pickle have the same amount of sugar? Did every pickle have the same amount of salt particles on it? Could that make a difference? If one person took a bite of the salty pickle and they're like, ugh, right? And another person was like, yeah, this is amazing. Maybe it wasn't the salty pickle. Maybe it was the amount of salt, okay? So what I'm trying to explain here, guys, is that when you go to do an experiment, it's super, super important that all the variables are the same. So you don't want to change everything up. Everything needs to be controlled. Everything needs to be constant. Everything needs to be the same. So there's only one variable that you're testing, which is my ingredients. That's it. It's the only thing that's going to be testing. So I need to keep the size of the pickle the same. The amount of the ingredients the same. The surrounding temperature needs to be the same. Okay? Now, once I have my data, I'm now going to get into my analysis. Now, one thing I want you guys to write down is a word called opie, he, act. So this is like a synonym, I guess. I'm not even an English teacher. So it's just an easy way to remember the scientific method. Let's put it like that. So opie, he, act. So I want you guys to bang on your head all day for the next couple of days. Opie, he, act. Opie, he, act. Okay. O, observation. P, problem. So opie, I is background information. He, opie, he. H, we made our hypothesis. E is our experiment. So opie, he. Okay. Act is the last part of the scientific method. So A, C. Okay, so act is analysis and then conclusion. Now when you guys go through your analysis, what you're going to do at home, okay, is that you're going to take the data that I'm giving you from the Pickle Lab and you're going to formulate a graph. And again, go back and look at the shark data. Go back and look at your lecture on graphs. How do I set up a graph? Don't forget, there's a couple of variables here. Variable number one is please make sure to include the dependent data. Okay, so dependent data, dependent variables. Dependent are our rate of taste. So our rate of taste was, it was horrible, number one. Number two, it was okay. Number three, it was interesting. Number four, man, that pickle was awesome. Okay, so our rate of taste dependent variable is going on the y-axis. Okay, now what goes on the x-axis down here is your independent variable. The independent variable is what I, as a researcher, I as a researcher chose to study. I chose sugar. I chose salt. I chose chocolate. Okay? So all of those things are going to go down here on the bottom, including your control. Okay? So now as scientists, you have to include the control in your data so we have something to compare it back to. You guys, if everyone had all this awesome data about sugar and salt, we cannot make a conclusion if we don't know what the normalcy is. Okay? So include your control. What did everyone think of butter pickles to begin with? Okay, what do they think about it in the end with the sugar and the salt and the chocolate? Now I'm going to be asking you guys a couple different ways. You can either graph everything independently, which means that you're going to have 5 separate graphs with all this data, or instead of wasting all that time, why don't you add up the numbers in the column of sugar for all the different people and give me an average. What was the average person's taste for the sugary pickle? One set of data, super easy. So now you only have 1 bar on 1 set of data and you can actually analyze the information, okay? So 1 bar for sugar, 1 bar for salt, 1 bar for chocolate, 1 bar for your control, and you're just going down the line of your data, getting the average, what everyone thought of the control, what everyone thought of the salt, all right? Okay, now please keep in mind, and again, this should be a review, but keep in mind when you go to write your graph, hand draw it, however you guys are going to do it, you need to include a title, okay? So the title to have in it, not just Pickle Lab. Okay, that's great back in 7th grade. Okay, you guys are now in honors bio in high school, and so the title needs to be extremely specific. The title needs to include what, where, when, and who. Okay, so what was the study on? Let's say great white sharks. Where was the study? New Zealand. So great white sharks analysis in New Zealand. When was it done? October 2019. By who? By Dr. Jenny Chambers. Okay, so what am I doing? Where is this happening? When did this happen and who did it? Because all of that helps to validate the science, validate the evidence. So if you're saying, oh yeah, this was done by Pop Sugar, right, back in 1975, right, you're thinking to yourself, why am I using this data? Because Pop Sugar isn't a valid resource, and 1979 is way too old to compare what's happening now. All right, so as scientists, we want to include that in our title for the researchers that are looking at our experiment. Now, the last thing you guys are going to do is write a conclusion. Now, the conclusion, and again, we will be going over all of this at a later time on how to type this all up, so don't worry about that. What I want you guys to do is just handwrite everything, draw everything out by hand, totally fine. Okay, the last thing you want to do as a conclusion is to tell the reader what you thought happened during the study. And you want to use facts and evidence. You don't want to use your personal opinion. You don't want to say, well, these guys thought. No, I don't care about that. What I care about is the data. Okay, so stick to the facts, stick to the data. Now, number one, when you go to write your paragraph, this is a paragraph written conclusion, you're going to include what did you do? And this is for all experiments that we're going to do in this class. So as you write your conclusion, tell the reader, this is what we did. We did a study on pickles. You're going to tell the reader what your original claim was. Okay? So it was believed that the chocolate pickles were going to be the best because who doesn't like chocolate? Everyone in the world loves chocolate. So chocolate and pickles should go hand in hand. You're then going to give us the data in words, in sentences. Don't just show us a picture. In words and sentences, write out what you found in your graphs, in your data tables. Give us a word, saying, sentence, whatever, that explains what you guys actually found. Most importantly, though, are the last two. Okay, so as a researcher, I'm going to be reading your data on your study, and I'm going to say, okay, awesome, this is what they found, I can see that in your graphs, this is what you did, this is what you thought was going to happen, but how did it work? How did it actually work out for you guys? So you need to include in the conclusion errors to the study, okay? Not just, hey, this is what went wrong, I forgot to cut all the pickles the same sizes, but how did it actually affect your overall data? So if I'm getting an average taste test that the sugary pickles were the best, those were the best. Well, what happens if those pickles were bigger than the pickles that I put salt on? Or what happens if those pickles were smaller than the pickles that I put the chocolate on? Is that going to make a difference? Okay, so you want to tell us maybe how this error might have affected your data. And the last thing I want you guys to do within the error section is to tell me how to fix it, okay? So next time this study is performed, it is recommended. Don't put your name on it. Don't say you or we or the group, okay? It's just third person, the researchers found that it is recommended to, okay? So just keep it very plain. We don't want to point fingers at any one person, race, gender, age, or whatever. It's science. It's all about the facts. So it is recommended that, blah, blah, blah. The last thing you want to do in your conclusion is to tell me why you did this in the first place. How does this even matter? Who cares about pickles? Okay? Tell us about it. This is your real world connection. Now, you guys, you're in honors bio. This is critical thinking. You're gonna have to think outside the box on this. So take a second, stop the video, and just think for a minute. How would you connect a pickle study on salt, sugar, and chocolate to the real world? Stop the video. Okay, now, hopefully you guys wrote down some ideas. This is my idea. It's not the only one. You guys could probably have a million more that are just as valid. The real world connection is money, okay? So we have a specific brand, Mount Olive. They're trying to be more competitive against maybe some of these other pickle companies that are coming out. And they want to sell their brand more so by changing up the recipe. So as a researcher, you're going to say, well, you know what? This matters because this company could go out of business if we don't switch up the recipe. And guess what? On a study of 100 people testing these different ingredients, it was found that the sugary pickles were the best. So if we add a little bit of sugar to this ingredient and call it sugary butter pickles, okay, or butter chip sugar, whatever you guys want to do, all of a sudden the company's going up, we're making more money, we can hire more people to the company, so now we have less poverty, less people on the street, less homeless, which means that you don't have as much disease that's going on between people that don't have money to go to the doctors. People are healthier, they're happier because we got money, we got great pickles, and suddenly we're changing the world, guys, because of sugary pickles. Are you with me? All right. Hope you guys enjoyed this quick lab on pickles, right? Please keep in mind, opihiat. All I need for you guys to do, as a reminder, is just write this whole thing down. Observation, problem, hypothesis. Give me your experiment data that was collected. Draw out a graph for your analysis. Include a title. Label your dependent, independent variables. Include your key, right? What is 1, 2, 3? Horrible, terrible, awesome. And then, of course, make sure to hand write a conclusion. And I'll be telling you guys what to do next after this. Hope this was entertaining and hope this was helpful. This is Holland Chambers Biology. Checking out.